Okay, let's dive into this explainer. Today we're looking at something literally mind-bending, the invisible molecular machines that make life possible. We're talking about enzymes. Now you hear that word all the time, right? Like in commercials for laundry detergent or fancy health supplements. But the real story, it is way more fascinating. Enzymes are the absolute ultimate biological speed limits. Without them, the incredibly complex, highly ordered processes happening inside our cells right now would just stop.
Seriously, think about this for a second. What if digesting your breakfast took years? I know, it sounds totally absurd, but chemically speaking, the breakdown of something like sucrose, just everyday table sugar, into carbon dioxide and water is actually thermodynamically favorable. It naturally wants to happen. But it is unbelievably slow. Complex biological molecules are kinetically stable. Without a catalyst, that sugar would just sit there for years before breaking down. So, without enzymes to speed up these specific reactions, basic digestion would completely stall out. Your muscles wouldn't contract on cue, and life as we know it simply couldn't exist.
To really wrap your head around the staggering scale of enzyme power, take a look at this number, 10 to the 17th power. That's a 1 with 17 zeros. That is the rate enhancement achieved by an enzyme called arotidine phosphate decarboxylase. It's an enzyme absolutely vital for making DNA and RNA building blocks. Get this, an uncatalyzed version of this exact reaction has a half-life of 78 million years. Let that sink in, 78 million years. But with this enzyme, the reaction happens in mere milliseconds. No way, right? But that is the kind of breathtaking acceleration we're dealing with.
So how exactly do they pull this off? Well, let's animate enzyme action. Substrate binds to the active site, forms an enzyme-substrate complex. Catalysis occurs, products are released, and the enzyme remains unchanged. It's this precise, beautiful, microscopic loop. Basically, the substrate, which is just the specific molecule the enzyme acts on, collides with a highly specialized pocket. They lock together, chemical bonds are instantly broken and formed, and the brand new products pop right out. And the best part? The enzyme is left perfectly intact, totally ready to just grab the next molecule and do it all over again.
Now, what's really interesting here is seeing the actual physical reality of that active site. We're looking at the structure of chymotrypsin. It's a digestive enzyme. See that highlighted red area? Those are the key amino acid residues, perfectly arranged by evolution to create a fully customized pocket. It's not just some flat surface. It is a three-dimensional cavity that perfectly encloses its substrate. It essentially hides it away from the chaotic, busy environment of the cell so the chemical reaction can happen under perfectly controlled conditions.
But here's an absolutely crucial point. We really need to understand what enzymes actually change and what they don't. An enzyme does not change the final equilibrium of a chemical reaction. Like if a reaction converts molecule A into molecule B, the enzyme isn't going to alter the final ratio of A to B. What it does is dramatically, and I mean dramatically, speed up the rate at which that equilibrium is reached. It basically takes an incredibly slow, difficult dirt road and turns it into a molecular superhighway.
And they build this superhighway by tackling something called activation energy. Think of this as a massive chemical energy barrier. And here is the truly wild part. You actually want this barrier to exist. Seriously, if it didn't, the complex macromolecules that make up your body right now would just spontaneously break down into simpler forms. Poof! We literally rely on this barrier for our structural stability. So what enzymes do is selectively lower this barrier, but only for the specific reactions the cell actively needs to happen in that exact moment. If we compare these two paths visually, the trick becomes pretty obvious. That higher curve is the uncatalyzed reaction. It's got a massive energy hill to climb. But the lower curve? That's our enzyme-catalyzed path. Notice how much lower the peak is? The enzyme actually shrinks that activation energy peak using something called binding energy. See, when the enzyme and substrate form weak, non-covalent interactions, things like hydrogen bonds, it actually releases a little bit of free energy. And that released energy effectively pays the thermodynamic cost to lower the hill. It makes the reaction vastly easier to achieve.
To make this a bit more intuitive, researchers use this brilliant thought experiment called the stick-a-days analogy. Okay, imagine you want to break a metal stick. The exact moment the stick is completely bent and stressed right before it snaps, that is our transition state. Now, if you had an enzyme that perfectly fit the normal straight stick, well, it would just stabilize it. The stick would sit there happily forever, hopelessly stuck in its comfortable straight stick pocket, and it would never break. So, to be an effective catalyst, the enzyme must be perfectly complementary to the bent stick. By forcing the substrate to fit into a bent-shaped pocket, the enzyme literally pulls it into the transition state, making it incredibly easy to snap.
And this brilliantly illustrates another wild thermodynamic trick enzymes have up their sleeve, and it involves water. Inside a cell, both the enzyme and the substrate are basically surrounded by a highly ordered shell of water molecules. When the substrate enters the active site, those water molecules are displaced. They get released out into the wider cellular fluid. Now, this release creates a massive increase in disorder, or entropy. And from a thermodynamic perspective, the universe absolutely loves an increase in entropy. So this process provides a powerful thermodynamic push that practically drives the substrate right into the enzyme's active site.
But wait, the enzyme isn't just some rigid piece of a puzzle waiting for another piece to fall in. Back in 1958, a scientist named Daniel Koshland introduced the concept of induced fit. Before that, we used to think of enzymes and substrates strictly as a rigid lock and key. But really, it's way more like putting on a comfortable glove. When the substrate binds, the enzyme dynamically molds and shifts its shape right around the molecule. It's a whole network of coupled motions that perfectly aligns all the chemical groups needed to trigger the reaction. A perfect real-world example of this induced fit is hexokinase. It's an enzyme that adds a phosphate group to glucose. When a glucose molecule enters its active site, the two main domains of hexokinase literally snap shut around it, just like a pair of jaws. Bam! And this conformational change is absolutely critical. Not only does it position the glucose perfectly for catalysis, but by clamping down so tightly, it completely locks out competing water molecules that would otherwise ruin the reaction. It is just a highly sophisticated microscopic machine in action.
Okay, so with thousands of these incredibly fast machines operating inside a single cell at any given moment, how do we prevent total chemical chaos? Well, the answer is exquisite regulation. The cell actively controls these enzymes. It basically acts like a chemical switch, turning them on or off by sticking chemical tags onto them, a process known as phosphorylation. Or it uses shape-shifting modulators from afar, where molecules bind to the enzyme and change its structure remotely. Sometimes, the cell even produces inactive precursors that have to be precisely snipped and activated exactly when and where they're needed, like in your digestive tract or during blood clotting.
Which brings us to a final, pretty awe-inspiring thought. Every single time your heart beats, every time you take a breath, or even form a thought about what you're learning right now, it is entirely dependent on thousands of these precisely regulated, unbelievably fast reactions happening millions of times a second. So how do microscopic, completely unthinking strings of folded amino acids manage to seamlessly orchestrate the breathtaking complexity of the human body? It really is a testament to the sheer power of evolution and the unbelievable elegance of biochemistry. I hope this explainer gave you a whole new appreciation for the molecular machinery running inside you right now. Keep wondering, keep learning, and I'll catch you in the next explainer.